Hi, my name is Lillian Murphy, and I'm an undergraduate senior at the California State University, Sacramento in the Department of Biological Sciences. And I'm currently working in Dr. Kimberly Mulligan's lab to identify environmental chemicals that impact neurodevelopment. So specifically, I'm here to talk to you today about how the exposure to hydroxylated polybrominated diphenyl ethers, or PBDEs, impairs axon pathfinding in wild-type Drosophila melanogaster, or the common fruit fly. And here you can see on the slide a picture of what a fruit fly looks like. So their neurodevelopmental disorders affect more than 1 in 10 children in the United States, and they are a group of heterogeneous disorders that range from motor disorders to intellectual disability, autism spectrum disorder, or ASD, and ADHD. And although there are known genetic components to neurodevelopmental disorders, it is thought in the field it is a combination of genetic factors as well as environmental factors coming together. And so some environmental factors include drugs, hormones, maternal autoantibodies, and the group I'm talking to you today about, environmental chemicals. So there's over 80,000 chemicals in the EPA inventory where there is little to no information on how they impact our health. But there are some chemicals that have been previously associated with neurodevelopmental disorders due to research, including methylmercury, BPA, polychlorinated biphenyls, or PCBs, lead, and organic phosphate pesticides. But the group I'm talking to you today about is PBDEs, or polybrominated diphenyl ethers. And they are a persistent environmental chemical where there's 209 conjugures, or 209 variants, that began being used in consumer products in the 1970s. Specifically, they're found in flame retardant consumer products, including textiles or children's pajamas, electronics, and polyurethane foam items such as couches and mattresses. And so how they enter the environment is because they are not chemically bound to these consumer products. And thus, when the products begin to degrade, then these PBDEs are released into the environment. And so exposure routes include dust exposure. So you can imagine a little child crawling around with PBD dust on the floor, dietary intake, including dairy products and seafood, and samples of air, soil, and water have found PBDE concentrations in them. And furthermore, their structure is easy to pass through the placental barrier and thus pose as a risk for toxic exposure to developing fetuses. Previous studies have found that they interfere with the cognitive function and behavior in animal models, and that children exposed to PBDEs have a decreased IQ level. There is one that is most prevalent in the environment and in human samples, and it is PDE47, and it has been studied to have toxic effects, including it disrupts the hormone or the endocrine signaling found in humans. And so you can see here, it's a structure of PDE47, and it's similar to hormones that are naturally occurring in our body. It also causes irregular calcium levels in the cells that are prior to neurons, that are neural precursor cells. And it alters the epigenetic patterning, or changes the amount of methylation found on DNA. So it increases it to cause a decrease in which results in a decrease in gene expression. Furthermore, although it is PDE47 is associated with neurodevelopmental disorders, it is unknown how these molecular dysfunctions, or even more, could be potentially linked between NDDs and PDE47. So even more to know about is that PDE47 in our bodies can be metabolized using this enzyme called cytochrome P450 that's naturally occurring. And this enzyme transforms it by adding a hydroxyl group at either the 6-OH or the 5-OH. And so from now on, I'll be referring to this as 6-OH and 5-OH, respectively. And so furthermore, this 6-OH compound has been studied and found that it disrupts the transcription of genes associated with neurodevelopmental disorders, and it alters the chromatin remodeling, or alters how the DNA is packaged within cells. So the goal of my project was to look at how 5-OH, 6-OH, and PDE47, or the parent compound, affect neurodevelopment using a model organism called Drosophila melanogaster, or the common fruit fly. So we expose our flies to the chemicals and then look at how the nervous system has developed. And so here we can see a figure of a neuron. And so we're particularly interested in the axon, which is this portion. And the axon is important to allow neurons to reach their target cells. So they have to grow in a precise manner in order for proper neurocircuitry of our brain, and impairments of this growth are linked to neurodevelopmental disorders. So our overarching question is, how do PBDE metabolites disrupt neurodevelopment in vivo or in our model organism, Drosophila? So we begin by exposing our flies to the chemicals by mixing it into their food. Then we introduce the flies, first the females to ensure embryonic exposure, and then males. And then we dissect the brains of their 0 to 2 day old adult offspring. We then do a protocol called immunohistochemistry, or IHC, where we take the adult Drosophila brain, as we see here, and we use antibodies to specifically tag the center or a portion of their brain called the mushroom body, which is the center for olfactory-based learning and memory. And in order to visualize this structure, we use fluorescent antibodies that specifically target this area and the previous antibodies we used. And so we visualize this by using confocal microscopy. And so we put hole-mounted brains on slides, and our confocal microscope will go through and take sections of the images throughout the brain. And we want our target image to look similar to this schematic here, where we see the mushroom body in green, and it's a bundle of neurons that have this particular shape. And so here's a bigger picture of it, where we see this bundle, it has these L-shaped, and there's no neurons or axons that have projected across the midline. And this is the proper phenotype of this mushroom body structure. However, there's different categories of defects that we see when we expose our flies to the chemicals. One is mild, where it's a thin band of crossing. Moderate is thicker, but not a full fusion. And severe is a full fusion of these neuron bundles together. So our results indicate that our 5-OH sample and our parent compound of PBE47 have a roughly 40% frequency of crossing or axons crossing the midline inappropriately. And then our 6-OH sample had a higher frequency of around roughly 60%. And this other chemical we tested called PD had a similar frequency of 60%. So I'll come back to explain what PD is. But first, I wanted to explain that we did a statistical analysis to see if there was a difference in the frequency of crossing at a statistically significant rate. And so we turned our qualitative data into quantitative data by saying our no crossing is a 4, down to severe crossing is a 1. And so our one-tailed Mann-Whitney statistical test showed that our 6-OH sample and our PD sample had a decrease in effect score, meaning that they had more frequency of crossing in the mild, moderate, and severe ranges. And thus, we see our statistically significance less than 0.05 p-value. So overall, we see that the 6-OH compound impairs axon pathfinding in a similar frequency to a known MAP kinase inhibitor. So I'm coming back to what PD is. So PD is a known MAP kinase inhibitor, and you might be asking, what does MAP kinase mean? MAP kinase is a pathway within cells that regulates many different cellular functions. In particular, the MEK-ERK signaling pathway, a type of MAP kinase, regulates the development of axonal growth, which is exactly what we're talking about and studying in this assay. And so here you can see what the signaling pathway looks like, and it's important for the expression of many different target genes. And so in particular, we find that our 6-OH flies also had a decrease in ERK's expression compared to our control flies. And this project was in collaboration with a group from UC Merced, and they were looking at this 6-OH compound as well as the other chemicals using a neuron cell culture. And so I wanted to share the results with you to give you a more complete picture of this study, where we see that they found the 6-OH inhibited MEK-ERK signaling and gene transcription in neuron cell culture. It also suppressed the neuronal electrical activity, so the neurons were not functioning properly, and it acted as an ATP kinase inhibitor, meaning that it inhibited many different kinases throughout the cell. And so all of this was actually summarized in a paper that was recently accepted for publication in the Journal of Biological Chemistry. And so overall, we helped uncover a new toxicity mechanism of PBDE metabolites impairing neurodevelopment, and that indicates that these flame retardant chemicals should be further studied as how our bodies can metabolize them and turn them into more toxic chemicals, and that these metabolites should be further studied in rodent models to see how they impact neurodevelopment and human health. I would like to thank my PI, Dr. Kimberly Mulligan, our collaborators at UC Merced, as well as our funding agencies, the NSF and the NIH, specifically the LSAMP and RISE programs. And thank you for your time.